Perfect. So welcome to week 2. We're going to start off with central dogma.
Yep, just give me a second. Yeah, so central dogma. So DNA structure. So this should be something that you should have either covered in 3-4 bio or during the bridging course, but just for some basics, DNA is made up of nucleotides. It's the deoxyribose sugar and the phosphate molecule that creates the backbone. And then you have the 4 different types of nitrogenous bases, adenine, guanine, thymine, and cytosine. Yep, next slide. Yeah, so that's just like that. What's in that little square there is considered one nucleotide. Yep. So in terms of the key features of DNA and what really separates DNA from other molecules is its double-stranded nature, the uniform diameter, the right-handed twist, and its anti-parallel strands. So this becomes quite important when we get to DNA replication because DNA can only be read from a 5' to a 3' direction, and nucleotides can only be added to the 3' end. So that sort of affects the way that DNA replication can occur. So if we move on to DNA replication now, just very quickly, these are the structures that actually allow the DNA to sort of fulfill its function in the cell. So this is a pretty good table here. So the 4 main functions of DNA is to store genetic material, to be able to be replicated. It should allow for gene expression, and it's susceptible to mutation. And you can see on the right side of the table there the features that allow that to happen. So DNA replication. Yeah, okay. So DNA replication is almost completely based on the complementary base pairing of adenine to thymine and cytosine to guanine. And so this is something to remember for when it comes to exam time. They like to use the term semi-conservative, which essentially means that one strand of the original DNA is maintained and then a new DNA strand is created. So every piece of DNA, every time there's DNA replication, it's semi-conservative. And that's just something to keep in mind when it comes time for exam preparation. And then, yeah, again, DNA grows from a 5' to 3' direction. And there's a sort of diagram coming up that will show that in more detail. Just quickly, in terms of prokaryotic and eukaryotic DNA replication, this isn't particularly important when it comes to the central dogma of humans as such, but the difference between prokaryotic and eukaryotic DNA replication is the fact that prokaryotes only have 1 point of replication, whilst eukaryotes have multiple, and this is a result of having such a large amount of DNA to start off with. And this will sort of be a little bit more relevant when it comes to sort of biotech and PCR and that kind of stuff. So if we go to the process of DNA replication. Okay, so in the process of DNA replication, there's basically 2 subsets. There's the leading strand and the lagging strand. So if we were looking at the leading strand, those are the steps there. I don't think there's very much point in me reading through them. But I think the more complex part of DNA replication is the lagging strand. So if we go to that slide, which is the next one. Okay, so the lagging strand is basically different to the leading strand because like I explained before, the fact that DNA can only be read from a 5' to 3' direction. And basically the lagging strand is a result of the anti-parallel nature of DNA. So what's the main difference is the fact that the DNA is unwound and that's the same, but there needs to be primers that are made in little bits through the DNA, and they eventually form the Okazaki fragments, which are the actual DNA molecules that are being created to be complementary to the initial strand. And then they're joined together by ligase. And that's a little bit easier to understand when you look at the diagram, which is on the next one. Yeah, okay. So the top strand here is the leading strand, and you can see on that strand, everything's quite simple. There's the initial primer, and then DNA polymerase just creates the remaining DNA, and then it's that primer and the DNA are joined by ligase, and that's pretty straightforward. The lagging strand, because it's going in the opposite direction, you can see those little red bits there. They're called primers, and they're created by primase. And that's essentially so that the DNA polymerase has somewhere to bind to and create those little Okazaki fragments. And so basically during the process of DNA replication in the lagging strand, there'll be a whole bunch of primers that are created, and then they're eventually replaced by DNA again, and then those Okazaki fragments plus the new DNAs are all joined together by ligase. Does that make sense? Again, if anyone's got any questions, please feel free to chuck them in the chat. But okay, we'll move on. So this here is it's just a table of all the enzymes that are used in DNA replication. It's just a quick and easy way to sort of remember all of them. Moving on to the next slide. Okay, so errors and repairs. This is again something that comes up a little bit during exams. They like to ask, it'll be maybe like 1 or 2 questions during the exam. So there's basically 3 types of repairs that can occur during DNA replication. There's first of all proofreading. So that's basically the repair that happens during the replication itself, and this usually fixes like 99% of the errors. And then there's the mismatch, which is the type of repairs that happens immediately after replication. And then there's excision, which happens over the sort of the lifespan of the cell, and that can help with fixing errors that are the result of chemical or radiation damage. Yeah, moving on. RNA. Okay, so RNA really is really similar to DNA. In terms of the actual makeup of RNA, the only difference is instead of the deoxyribose sugar, there's a ribose sugar, but everything else is the same, except for in terms of the nitrogenous bases. The thymine that's found in DNA is replaced by uracil in RNA. And then the other thing is that RNA is a single-stranded molecule. And then there are a couple of different types of RNA. So if we go on to the next slide, there's a table there. Yeah. So there's messenger RNA, which is used to code for proteins. Ribosomal RNA is what's basically creating, that allows for the synthesis of proteins. Transfer RNA is what carries the amino acids to the ribosomes during protein synthesis. And then there's pre-mRNA, which is sort of a subsection of mRNA, and that's what's produced at the end of transcription. Okay, so these are the 2 major steps of protein synthesis. There's transcription and then there's translation. And they both sort of occur in different places in the cell, so we'll go through them both individually. So if we go through transcription first. Okay, so transcription is essentially the production of mRNA. So it happens in the nucleus, and it's the conversion of the DNA or the gene to pre-mRNA to mRNA. So if we were to look at the steps more precisely, the transcription factors will bind to the specific sequence of DNA that is coding for the protein that we want. And then the RNA polymerase will basically create the RNA strand that we want, growing from 5' to 3', so similar to DNA in that way. And then it will continue to do, the RNA polymerase will continue to do so until it reaches the terminator section of the gene. And that is what we call pre-mRNA. And then if we were to go to the next slide, so that's the production of the pre-mRNA. Next slide. Okay, so basically post-transcriptional modifications is what converts pre-mRNA into mRNA for that mRNA to then be sent to the ribosome to actually produce proteins. So there's a couple of different post-transcriptional modifications. There's the methyl G cap that is added to the end of the 5', the 5' end of the mRNA. There's a poly A tail at the 3' end, and then there's splicing. So generally speaking, splicing is the splicing out of introns to leave exons. But I believe that you guys will have a workshop or a tutorial of sorts where you'll talk about alternative splicing and a few other types of splicing. But essentially, splicing is when they take out the parts of the gene that they don't want and leave in the genes that they do. And so now basically the mRNA is ready to be exported from the nucleus into the cytoplasm to the ribosome. So and it's ready for translation now. Yeah, okay. So translation is the conversion of the mRNA to a peptide chain. And it occurs at the ribosomes that are in the cytoplasm. Or it can also occur at the ribosomes that are on the rough endoplasmic reticulum as well. So quickly, before we can actually go through translation, we have to sort of understand the structure of a ribosome. So the ribosome is essentially made up of 2 subunits, and there's 3 sort of parts. There's the A site, which is where the amino acid that's matching the codon on the mRNA is brought by the tRNA, and that's where they first sort of make contact. And then the P site is where the peptide link is formed between the 2 adjacent amino acids. And then the E site is when the uncharged tRNA, which initially carried in the amino acid, will leave. And there's a picture in the next. Yeah, okay. So you can see the A site, the P site, and the E site there. So I think a really good way to think about it is like a ribbon that's being pulled through a sort of strange burger sort of thing. And at each point, there's a specific role. So yeah, I've just summarized them again in that little text thing there. If we move on to the actual process of translation, there's 4 basic steps. There's the initiation, which is essentially when the first codon, which is the start codon, enters the P site. Elongation is when the mRNA slides into the A site, and then charged tRNAs bring the necessary amino acids with the matching anticodon to the bring the thing to the ribosomes, and the bond between those 2 is what's formed in the P site. And then termination is when the stop codon enters the A site and is recognized for a release factor, and the polypeptide chain is released. And then after the polypeptide chain has been created and released, it can undergo a few changes and modifications, which can include localization and export. It can undergo more folding, and it can also undergo some regulation and stuff along those lines. I hope that made sense. If you guys had any questions about that, please feel free to chuck them in the chat.
All right, I think this is me. So I'll be covering cellular energetics this week. Next slide, please. Yep, so this is just some prior knowledge. So you might have learned it in biology or even in chemistry. A lot of you probably would have done chemistry. So basically proteins, they are polymers of covalently bound amino acids. So these amino acids basically join together to form proteins. So there are many functions of proteins. Some of the main ones are catalyzing reactions, movement and stability. So you might see them in the cell membrane, defense, signal, storage, and transport. So there's the three main structures of a protein: primary, secondary, tertiary, and quaternary. You can read this in your own time, but yeah, you probably might know them already. Next slide, please. Yeah, so cellular energetics. So energy is basically the ability and the capacity to do work. So the capacity for change. So what do we know metabolism. Metabolism is basically the sum of all chemical transformations that occur in the body. So it's a result of atoms having enough energy to combine and change their bonding partners. So metabolic reactions, they are energy changes which are associated with change in chemical composition. So with energy, there are two main types of energy. There's potential and kinetic. So potential is basically that energy that's stored in chemical bonds, concentration and electrochemical gradients. And kinetic energy is the energy of motion. So when you're moving, that's when you have kinetic energy. So as you see on the cat on the side, when she's resting, she basically has that potential energy, which is then converted into kinetic energy once she is active. Next slide, please. So metabolism, so continuing, there's two types of reactions. So there's catabolism and anabolism, actually, whichever way you say it. So for catabolism, that's the breakdown of large molecules. So you're starting off with big molecules and you're breaking it down, whereas anabolism is having small precursors, small molecules, and you're basically trying to make larger macromolecules. So it's the opposite way. So in that table really summarizes both of them. So with catabolism, energy is released. So basically you're breaking down the large molecules, you're releasing energy. And for anabolism, you're using energy. So you probably think of it that way, that you're trying to make bigger molecules. You probably need more energy and more resources to be able to make those big molecules. And that's why you need to use energy. And anabolism is basically reductive and catabolism is oxidative. And in catabolism, you have the release energies restored in new bonds, or it can be used as kinetic energy. And in anabolism, as we said, it's producing complex small molecules from small precursors. Yep. All right, so the law of thermodynamics. So this is good to know. So first law of thermodynamics is that energy is neither created or destroyed, and the total energy in a reaction is always the same. And the second law is that when converted from one form to another, some energy is unable to do work, so it's lost to disorder. Next slide, please. Yep, so entropy. So this might be a bit confusing to understand. I found it a bit confusing, but it's basically a measure of disorder in a system. So catabolic reactions, they have increasing, higher entropy. That's because the complexity decreases. And anabolic reactions, they have decreased entropy because the complexity increases. So catabolic is basically breaking it down, as we said, breaking down bigger molecules. So you're decreasing the complexity. So you have increased entropy, and anabolic is basically the opposite. So delta G, if it's negative delta G, then there's basically free energy released. And if you have positive, then you have free energy required. So that's anabolic, and the other one is catabolic. And then when you have 0 delta G, that basically means that the reaction is at equilibrium. Next slide, please. All right, energy coupling. So basically this happens when energy produced by one reaction, it's used to drive another reaction. So one reaction releases energy and another reaction is able to use that energy. And ATP is intermediate coupling for anabolism and catabolism. So the hydrolysis of AT, that's when you remove the last phosphate group, as you can see on the image on the right. You remove the phosphate group, and that converts it to ADP. And this process releases energy, and that's energy we can use in different reactions and processes. And these are called coupled reactions. And yeah, the reverse synthesis of ATP from ADP, so the opposite of that, you'll need energy. So that's anabolic. Next slide. Yeah, so enzymes are basically biological catalysts. They're proteins and they speed up the rate of reactions. Now, how do they speed up the rate of reactions? So as you can see on the image on the right there, the EA right there is the activation energy, and that's the energy barrier that's needed for the reaction to occur. And what enzymes do is that they reduce the activation energy barrier needed. And that means that the reaction can happen at a faster rate. So enzymes, they're not altered by the reaction, and the delta G does not change as a result of the enzyme. And enzyme activity can be influenced by its specific active site that is complementary to the substrate. And this active site either orientates to substrates to be in the right angle to react, physical strain, so the enzyme applies pressure to the molecules, and also adds electric charges. Next slide, please. So factors affecting enzyme activity. So there's 4 main factors. So first one is substrate concentration. So if there's too much substrate, then enzymes can become saturated, and that can decrease the activity or the rate of activity of the enzyme, because there's already many enzymes that are filled with substrates. They can't attach to new substrates. There's also regulatory molecules. So you have prosthetic groups, inorganic factors, and coenzymes. So some examples of them are at the bottom. So for example, some inorganic cofactors are iron and ions. And then you have coenzymes, which are NAD and ATP, and you also have prosthetic groups. You can also have inhibitors. So inhibitors basically that can stop or decrease the enzyme activity, and also gene expression regulation. So we know that enzymes are proteins, like I said before. So there might be fewer proteins being translated and produced, so fewer or more enzymes, and that can affect how often or how much we can increase the rate of reaction. Next slide, please. Yeah, so for enzyme inhibition, this is just leading on from the previous slide. You can have reversible or irreversible. Basically, what it means is that irreversible means that the molecule binds to the active site permanently, so it can't be taken off, but reversible means that it can be taken off. So you have competitive, uncompetitive, and non-competitive. So you can see on the image on the right, that might be easier to see what's happening there, but competitive is basically the molecule binds to the active site, so that directly blocks the substrate from coming on. Uncompetitive is that the molecule binds away from the active site, so the purple molecule on the side, and that prevents substrate binding as well, but it's not on the active site, so it's not directly competitive. And then non-competitive is that the molecule binds away from the active site, and it changes the active site shapes. As you can see on the last number D there, the active site has completely changed its shape so that the substrate can't bind. You also have allosteric inhibitors, so they bind to sites other than the active site, and they increase or decrease the likelihood of enzyme becoming active. Next slide, please. Yep, that's it.
All right, moving on to fuels. So do you want to stop sharing and then I can share my screen? Right, okay, I'll share my screen. Let's see. Right, so fuels, I feel like the title of the lecture is a bit misleading or like confusing, because it's like what does it mean, like what's fuels? But yeah, so like this is probably tomorrow's lecture, so unless you're really ahead, you probably haven't done this lecture yet. So what we're doing here is we're just going to give you an overview of this lecture, so what it's going to talk about is pretty much metabolism. So it's going to cover glucose metabolism and lipid metabolism. So it's mainly focused on glucose metabolism, and lipid metabolism, you will touch on it more next week as well. But this week, you will learn something called beta oxidation pathway, which is quite important. So all right, so this is basically an outline of what we're going to talk about today for this this segment. So we're going to cover glycolysis, pyruvate, like what's pyruvate, what happens to it, citric acid cycle, you might have heard of it, you might not, it's okay, it doesn't matter. Oxidative phosphorylation, can barely pronounce it. And then, yeah, so basically, like a lot of people find this very confusing. So a lot of people find metabolism and basically everything that happens around metabolism really, really confusing. But basically what you need to know is pretty much this diagram. So it's like you have these three things. You have glucose, you have fatty acid, fats, you have amino acid, which is basically protein. And then it all goes into, it all kind of produces acetyl-CoA. And then the acetyl-CoA goes into citric acid cycle. And then it goes into this thing called the electron transfer chain. And the whole purpose of all of these complicated processes is to produce more ATP so the body can have energy to function. And yeah, okay, I can't use them. Okay, so let's start off with glucose. So glucose metabolism. What is glucose? Glucose, basically just know that it is it's a carbohydrate. It's a type of carbohydrate, and it's very abundant. And we obtain it through eating food. For example, like I don't know, sugar and stuff. And yeah, the body is able to break down carbohydrate into glucose. And another way is to mobilize stored glycogen in our body. So like if you eat excess glucose, say you eat a lot of carbs, and then like the body will kind of store a part of the glucose away in the body, so like it can be mobilized when we need. And we do all of this just to get energy for our body. And yeah, the first step of glucose metabolism is called glycolysis. So if you look at the word, it's like glycolysis. Like lysis means like to, I don't know, I'm not good at English, but like break down or something. So basically what it means, like it converts glucose into a smaller molecule called pyruvate. So glucose has six carbons, and this smaller molecule pyruvate has 3 carbons. So because you need this balance, like you can't just randomly destroy a carbon. So we will have 1 glucose at the beginning, but like this 1 glucose will lead to 2 pyruvates. And this process, well, you don't need to remember any of these steps. You just need to know, like it starts with a glucose, it ends with like it ends with 2 pyruvates, and it also produces 2 ATP and 2 NADH. So I think before we get further into these steps, we need to kind of talk about what these things all mean. So like what's the ATP, what's the NADH? Like last year when I was learning, I was really confused about this stuff. So ATP, Mahek just covered a bit about it. So like basically it's like, it's basically a package of energy. So you just need to, like, you don't have to know what it is or like what ATP stands for, but just think when you see ATP, just think energy. So like the body gets energy from ATP. And NADH is basically, yeah, like just think that like NADH is kind of like, like it contains more energy, and when the NADH get converted to NAD+, it releases, yeah, I mean, you don't have to know like what it is, just like know that NADH has higher energy. And like the same goes for FADH2. So like these are all kind of like energy molecules. You don't have to know the details of it, just know that like these things kind of like are high energy, so it's like the body can use them. But pretty much like the only thing the body can use is like ATP. So like all of these things, the NADH, FADH2, GTP, they all need to be converted into ATP to like provide energy. But yeah, that's that. And moving on to the next step. So in glycolysis, we mentioned that we have like one glucose will produce 2 pyruvates and also like some energy on the side. Like then what happens to this pyruvate? So it's kind of separated into 2 scenarios. So like one scenario is you have enough oxygen, one is you don't. So if you have enough oxygen, the pyruvate will get converted into acetyl-CoA. And what acetyl-CoA does is basically acetyl-CoA can go into the next step, which is the citric acid cycle. But if you don't have enough oxygen, what it does instead is the pyruvate gets converted into lactate. And well, lactate can't go into the citric acid cycle. So basically pretty much your energy production just stops there. And so that's why if you don't have enough oxygen, well, you can still produce some energy, but that energy is entirely from the previous step, which is glycolysis. But if you have enough oxygen, then it can move on and to produce more and more ATP. Yeah, so like a few points to note here. So for aerobic respiration, which is pyruvate converted to acetyl-CoA, you need to have an enzyme called pyruvate dehydrogenase as well. Just know the name of the enzyme. And also a bit more about anaerobic respiration. So basically this is when pyruvate converts to lactate. So that's, if you look at this diagram, it's like if you, as we might remember from glycolysis, we produce 2 NADH. And here, like the NADH gets converted back into NAD+. And so if you have a lot of NADH, it would probably kind of, because of the equilibrium, if you did chemistry, which I don't really remember, but yeah, it's like it's if you have a lot of NADH, then it kind of just pushes the reaction in a different direction. So it kind of like pushes it like that way to produce lactate. And like the enzyme is called lactate dehydrogenase, and a buildup of lactate causes lactate acidosis, and that leads to kind of a low pH because like lactate is an acid, and to treat that you use the base, which is sodium bicarbonate. I guess this is not very high yield either. So, well, like it's just there for knowledge, and it was in the lecture slides from last year. Yeah, moving on to the next step. So this will be the 3rd step of glucose metabolism. So we get glycolysis. We have glycolysis, which is where we got pyruvate, and we have pyruvate converted to acetyl-CoA. And now the 3rd step is acetyl-CoA enters this circle called the citric acid cycle. It's also called the Krebs cycle, which is a term you might have heard somewhere else. Yeah, so it has basically just like a circle, and it has 8 steps, and it goes around and around forever. You don't need to know the steps, but just need to know the energy output. So like it produces NADH, FADH2, TDP, and lots of electrons. And the electrons will be important later on. Yeah, so right, moving on to oxidative phosphorylation. So the last step of this energy production. So oxidative phosphorylation is actually a name for 2 processes. So one is electron transport chain, and one is cumulosmosis. Yeah, so if you look at this diagram, so you can see like the electron transport chain is basically like you have 4 protein complexes that's embedded in mitochondria. So like this whole process is happening in the mitochondria, and these 4 complexes are in the inner membrane of mitochondria. If you remember from last week, your lectures on cell, you might remember the mitochondria has a double membrane structure. So it has the inner membrane and the outer membrane. So this electron transport chain is happening between the inside of the inner membrane, and also like the intermembrane space. So like that's between the inner and the outer membrane. And so these 4 protein complexes, what they do is they facilitate electron transfer. So if you might see on the diagram that like if you look at these energy molecules. So like the NADH, the FADH2, like when they're converted into NAD plus and FAD, like it looks like they lose, they kind of, they gain electrons. So like the electrons I mean, I actually don't need to know this in too much detail. So it's just like know where it is, know what happened, like know kind of energy is produced there. And so like this is just kind of like I'm explaining this just for like understanding, but it's like if you don't really understand it, it's like it's not the end of the world. And also you'll have lectures on this. So basically, this electron transfer is coupled with proton transfer across the membrane. So like the protons, so like you see the H+, so they are transferred from inside of the mitochondria to the intermembrane space. And because of this transfer of proton into the intermembrane space, there will be a lot of protons in that space. So like more than, so like that means they will have a high concentration of protons and also a high, a very high positive charge. So that creates what's called an electrochemical gradient. And this gradient, so like because it will be kind of like just to achieve a balance of like the concentration and electric charge, these protons will want to diffuse back into the mitochondria, so like into like the inner space of mitochondria, and they can do that through like basically another another channel, which is called the ATP synthase. So the ATP synthase, it basically, it's like when the protons are going through the ATP synthase, it pretty much turns the ATP synthase, or it's like it produces a motor force, and like because of the motion of the protons, and like that energy can be used to convert ADP to ATP. So yeah, like in the end, ATP is made, and then that means we've kind of packaged some energy for the body. Yeah, I don't know if that explanation makes sense, but it's like this is confusing for a lot of people. So like you're not, and like where's my diagram? Oh yeah, and basically in the end, like all you need to know is like the energy production, and like this table, it looks like there's a lot of stuff, but you don't even need to know all of these stuff. It's like you just need to know like kind of ATP is produced, that sort of thing. And also I guess a few things to take note of is like energy production and also the location, so like where these things are happening. Yeah, going back to my previous slides. So because we're still talking about glucose metabolism, right? And like on my first slide, I mentioned the like humans can get glucose from food, or we can get glucose from glycogen that's stored in our body. So how that's done is basically because yeah, like when I was looking through my notes, I found this this very bad this note with very bad handwriting, but I think it has good information, so I just put it here. So like the mobilization of glycogen happens when like we haven't got food for a while. So basically that means we can't get glucose from diet, so we had to get it another way. And the reason why it is important to get it is because glucose is an important energy source for the brain, and you all can talk more about this next week. And like if the brain doesn't have glucose, then it's not good when your brain doesn't have energy. So like we need to make sure that like there is kind of like a sort of consistent supply of glucose in the body. So yeah, basically the process is like you have this hormone called glucagon, and also epinephrine. Why am I calling it epinephrine? It's adrenaline, right? Yeah, so like basically it's like the body can sense your glucose level, and when your glucose level is low, then the kind of this hormone glucagon, like the level will increase, and the adrenaline will also that doesn't look right. But yeah, basically, all you need to know is like the body can sense the level of glucose, and when the glucose level is low, and it kind of like activates this pathway to break down the stored glycogen, and the glycogen will be broken down. Kind of the two main sites are liver cells and muscle cells. And yeah, so like in the liver, what happens is so it gets converted into this thing called glucose 6 phosphatase first, and then it like in the liver, what happens is it will kind of get converted back to glucose, and then like it goes through the glycolysis. And in the muscle, like the G6P molecule can be used directly and go into glycolysis. So like where this G6P molecule is like if you look at like between the number 1 and 2, so like that's where it is in the in the glycolysis process. Right. So like why? Yeah, I touched on why the mobilization of stored glycogen is necessary. So I guess we'll just so like that's everything from glucose metabolism, and we'll move on to lipid metabolism. We'll just like touch on this for a little bit because you will have a like more detailed lecture next week. So like this week, the most important thing to cover is pretty much just beta oxidation pathway. So like what that is right. So what beta oxidation pathway is, so if you have kind of the stored lipid in your body, and like that's how the lipid is like kind of metabolized and like gets broken down like in the end into acetyl-CoA. And so like what happens is like so basically the only thing you need to know is like it's, for example, like an example is if you have a fatty acid with 16 carbons, like the carbons get removed like 2 carbons at a time. And so that means like every time it removes 2 carbon, and like 1 NADH and 1 FADH2 is produced. And when these kind of like that, so like that means like energy is produced. And then in the end, you will end up with like 7 NADH, 7 FADH2, and like 7 proton as well. And like every step will produce like 1 acetyl-CoA, and the last step is kind of special. It will produce 2 acetyl-CoAs, but like you don't need to know like that much. You just need to know like acetyl-CoA is produced as well, and then like acetyl-CoA kind of like in the end they go into the citric acid cycle. So if we go back to our diagram somewhere, if we go back to this thing, like that is why I have this diagram here. It's like, so like no matter what it is, it might be glucose, it might be like fatty acids, like they all kind of, the metabolism all kind of converge when it produces acetyl-CoA, and like then this acetyl-CoA goes into citric acid cycle, and they all go through this same process. And I might just touch on a bit about the digestion and absorption of lipid from diet. How do I present? Okay, present. Right. So like the location of this, so like the digestion and absorption of lipid happens in small intestine, and like this is not very high yield. You pretty much don't need to know it, but like this is a pretty good diagram here. Like it's pretty much all here, but like the first step would just be like the dietary fat get emulsified. So like basically it kind of get packaged into like micelles. So like micelles are like little things like this, and then it kind of get hydrolyzed into fatty acid and monoglycerides. The kind of chemistry of this, well, you could look it up. It's like, yeah, it kind of gets separated into like a thing of like the fatty acid and then the monoglyceride, and it kind of enters the small intestine, like, and it goes into the cells, the intestinal epithelial cells, and like in the endoplasmic reticulum of that, it gets resynthesized into triglycerides, and this triglycerides get packaged into a thing called chylomicrons, and the chylomicrons go into lymphatics, and then kind of it gets sent like through the lymphatics, the triglycerides, the lipids get sent to different tissues. Yeah, again, it's not high yield, doesn't really matter. And I'll just touch on a bit about chylomicrons. Like again, next week you will learn more about this, but this thing is basically what's called the lipoprotein, and what it does is yeah, its job is just to transport the lipid from the intestine to tissues. Yeah, mobilization of lipids, so like that's kind of the reverse of the digestion. So like when these kind of like after the lipids kind of like you get the lipids through diet, and then kind of you digest it and absorb it, and it gets stored in your body. And when we need it, it's kind of the same as the mobilization of glycogen and glucose. When we need it, like it will just kind of get converted back into fatty acid, and like it goes into blood. And because yeah, like it in the blood is kind of attached to something called albumin, so like albumin protein, and it enters muscles, heart, wherever that needs it, and yeah, and then it enters mitochondria to go through beta oxidation pathway to be metabolized. Again, not high yield. I feel like every time I present, I just say everything's not high yield. But yeah, well, that's it. So let's move on. Do I stop share or do I keep sharing?
If you don't mind, Jufi, I'll be sharing my screen.
Okay.
Okay, so wait, give me a second. I lost where I was. Did I put lots of memes? Same amount as last time, but let's see how we go. So to be honest, guys, this is not like a really super heavy topic. It's not even as heavy as last week. There's basically 2 concepts to go through. So yeah, we're going through the national health system, which you all should know about. Okay, yeah, I did start off with a meme. So Australia versus US language differences. I'm just going to read this because I like reading in American accents, and Australian accents. So we have bloke, man, cyclone, hurricane, lift, elevator, shopping center, shopping mall, and we have universal healthcare, which they don't, so it sucks for them. So, sorry, I'm not really funny. Yeah, so structure of the health system. There's 2 different ways that I've seen we can express it in, but let's go through both of them anyway. So we have the 4 S's, which are stuff, staff, spaces, and systems. So they are the key components of any health system. Stuff is, like your hospital beds, your medical equipment, things that kind of you use in a medical setting. Staff, because we need people to run them. That's your workforce. Spaces, because we need places to run them in, hospitals, private practices, clinics, all of that. And of course, we have systems, which not just includes the national health system, but also like within each clinic or hospital, they have their own systems of running things, like, for example, how they prioritize patients, waiting times, things like that. That's all part of a system. Paperwork, that all comes under systems as well. And the other way of expressing the structure of a health system could be using twigs, which I got from last year's PSP. Anyway. So there's technology, which is same thing as stuff. We have workforce, so that's staff. So workforce can be split into primary, secondary, tertiary, and even quaternary, which I will be discussing in a later slide, so I won't go through it right now. But just so you know, quaternary stuff, quaternary workforce or quaternary medical care is basically when you, it's like the thing above hospital care. It's like when you try like experiments, like for example, I guess stem cell therapy, because it's kind of new, that could be considered quaternary care, something that, I hope I spelled that right, actually, not sure. Yeah, like it's basically beyond normal hospital care. And then we have infrastructure, which is your spaces. Wait, did I get that right? We have government and we have system. I need to double check actually G and S. I believe I forgot to check that. But bottom line of this is that I recommend going with 4 S's because I feel like that is the main way that UD teaches us. Bottom line is that health systems need to be regulated in quality and supported appropriately. We'll be talking about these support and regulation shortly. Australia operates under universal healthcare. Well, actually, it's technically mixed because we also use private health insurance, but yeah, we do have universal healthcare as well. In an efficient system like Australia's, patient agency and doctor empathy can go missing because like people get reduced to numbers and statistics, really. That's something you could talk about in class. All right, so health systems need 3 things. And I'm looking for 3 buzzwords here. This is a great opportunity for those who have already done the pre-reading for tomorrow. But can someone possibly tell me what those 3 things that health systems need are? Any volunteers? If you don't volunteer, I'm just going to randomly pick you, so you might as well speak up. We'll shout you out in our next post if you get any of these right. Let's see if there's anyone in the chat. Okay, we have someone saying patience, which is true. Does anyone want to speak, like unmute themselves?
Is it like regulation, I think, or something?
Regulation is correct. Let's see if that's there. Wait, sorry. Regulation, good job. Daniel, we'll be shouting you out.
Nah, please. I beg you. Please don't.
Okay. Whatever reward would suit you then. You can let me know. Regulation, what else? There's 2 more I'm looking for.
It's funding one, I think.
Funding is 1, correct? Correct. And the last thing, so we have the quality control, we have the funding to support it. What is the last thing that health systems need to be put into action? It's a D word. Money is, yes, funding, yep. Anything else? It starts with D. It's literally the thing that puts health systems out to the public. Like, what is the thing for? What is the word for it? Delivery, that is correct. Thank you to the anonymous person who DM'd me the answer. Yes, delivery, correct. So health systems need regulation, funding, and delivery so that you can deliver the services. All right, so regulation is first thing. Yeah, I did steal this straight from PSParachute. Really sorry, but I love them. Yeah, I'm not going to read this out. You can read it when I upload the slides to our PSParachute folder, drive folder. I will be focusing in on Medicare and PBS shortly, though. Yeah, you can read that in your own time. Okay, so funding. So funding of health systems is important because it leads to more money for the system, which leads to better resources and making it a better health system. And what does a better health system mean? It means you have better patient outcomes. So before I tell you the answer, can someone tell me what our 2 main sources of funding for our health system are? It's kind of obvious, actually. Can someone please unmute themselves? Well, okay, yeah, the government, yeah, right, the government does, yes, but what are the 2 schemes that the government funds the health system through? It's not like the government just randomly gives money, like which it can, but not always. Yes, that is correct, Susan. PBS and Medicare are 2 main methods of funding, which I did indicate in the previous slide. It was kind of a subtle hint, but not everyone got it. So let's go through Medicare first of all. So Medicare has 2 components in terms of like, wait, I'm just trying to remember what the 2 components are. Wait, let's just go through this. Okay, wait, so the 2 components are free treatment at public hospitals. And what is the other one? Can someone think of the other component? No? Okay, I'm going to tell you then. It's free or subsidized treatment by a Medicare provider number holders. Like some practices have like a provider number and then they can subsidize treatment for you. Okay, so Medicare is applicable to Australian citizens, New Zealand citizens in Australia, permanent visa holders, and reciprocal agreement country citizens. So you might not know, actually, does anyone know what reciprocal agreement is? Anyone? It's quite a novel concept, actually. Okay, well, I'll tell you. So reciprocal agreement is like Australia's made agreements with several countries. I noticed that a lot of them are in Europe, but several countries have an agreement that they can get free or subsidized healthcare when they come to our country and when we go to their country. So yeah, it's kind of like a cool agreement. Medicare charges a levy of up to 2.5% income to citizens, usually like slightly richer people. And additionally, up to 1.5% if you're not on private health insurance. So yes, the government does encourage us to take up private health insurance to fund some of that stuff. That's one way it does it. And this levy actually funds a quarter of our health system. Okay, MBS, which is the Medicare Benefits Schedule, is just a list of all services that the government pays a rebate to. So rebate is just kind of just like the subsidy that it pays. And subsidy for us, really. And doctors can charge more than the listed fee, like listed fees as in that the schedule has listed. So that means that the patient sometimes has to pay out of their pocket for that gap fee. So the difference between what the MBS lists and what the doctor charges. And on to PBS. So PBS, what does PBS stand for? Anyone? Unmute, please.
Is it pharmaceutical benefits scheme or something?
Correct. It is pharmaceutical benefits scheme. That's correct. So it makes drugs more affordable. Don't get excited yet. It also regulates the safety and quality of drugs. So no, ICE is not on the PBS. And Commonwealth government pays roughly 87% of the cost of drugs because they can get actually really expensive. And we pay the rest of it. Concessions are available for DVAs, healthcare card holders, and seniors or pensioners. And there is a safety net threshold, which is like, so after a certain amount, you either don't have to pay at all, like if you're a concession holder, or you pay like a post-safety net co-payment. So that's kind of like, yeah, that's just like after a certain amount, like a subsidy basically. And the third thing that I mentioned that is necessary for a health system is delivery. So delivery, we have primary care, which is the first point of contact, like your GP. Can anyone guess what the next type of delivery could be? It's really easy, I promise. It's really easy. What is after primary?
Secondary.
Secondary, yes, thank you, enthusiastic student. Yes, so we have secondary delivery, which is your second point of contact. Surprise, surprise, like your specialist referred to by a GP. So for example, dermatologist is a secondary point of contact. And we have tertiary delivery, which is your third point of contact or consultant specialist level care. If you're listening to me earlier, actually maybe I didn't say that earlier, but yeah, usually tertiary care is at a hospital. And there's coordinary care, but that's not that important for this. And that's all, folks. Just to recap, the most important things you need to know are how the health system is funded, so Medicare, PBS. Probably know a few of the things from that table I copy-pasted. And just know what primary, secondary, tertiary care are. And that's all, folks. And on to our last presenter, Yash.
Hey, everyone. So before we move on to the next slide, I just wanted to give a bit of a caveat that these these lectures on homeostasis and acid-base probably aren't the most high yield in terms of content. To illustrate this, I think last year our acid-base lecture was cut short by technical difficulties, and then we didn't actually get a lecture on homeostasis. So I think that pretty much demonstrates how sort of useful these might be in terms of exam preparation. But nonetheless, we'll run through some of the basics and things that could help you in the coming years or this year as well. So next slide, please, Sav. Okay, so I think the main thing to understand here is that at 37 degrees Celsius, which is our body temperature, the neutral pH in our body is 6.8, whereas the normal pH of the extracellular fluid in our body is 7.4, meaning it's a little alkaline. Now, normal blood pH must be maintained within a very narrow range, and that's typically from 7.36 to around 7.45. Sorry, it says 7.4 on the slide. And why is this important? Because it ensures that that there is a proper functioning of metabolic processes in our body and the delivery of the right amount of oxygen to tissues. So acidosis refers to an excess of acid in the blood that causes the pH to fall below 7.35. And alkalosis refers to an excess of base in the blood that causes the pH to rise above 7.45. And I guess clinically, many conditions and diseases can interfere with pH control in the body, and that causes one's blood pH to fall outside of these healthy limit. Now, something else to note is that the equation to figure out the pH is pH equals the negative log to the base 10 of the hydrogen ions. So yeah, I guess the other important thing to note here, which is pretty much the big takeaway from Professor Chris Wright's lecture, was that neutral pH is 6.8 in our body, whereas normal pH is 7.4. So there's a difference there. And I think when we were delivered this lecture last year, the question he told us was, do you think the blood in your body is neutral? And most people, raised their hand. But then throughout the lecture, he sort of explained why that isn't the case, and the normal pH of our blood is 7.4. And then you've got a bit of an equation here. So CO2 plus H2O gives hydrogen ion. So now the main thing to note here is that when CO2 rises, the level of hydrogen ion rises as well. And if CO2 drops, then the level of hydrogen ion drops as well. And pretty much the quickest way to quickly run through homeostasis, it's that homeostasis is the tendency of our body to maintain a stable or relatively constant internal environment, whilst the external environment sort of changes. And that's what we call homeostasis. So the body sort of responds to different stimuli. So yeah, that's a very big, a very quick run through of what we think is important for those lectures. And that brings us to the end. So thank you everyone who came today. And we might give a couple minutes for anyone to ask any questions. These don't have to pertain to what we've sort of covered today. It can be more generally as well about how maybe week 2 is going on and that sort of thing. But if not, again, thank you so much everyone for coming down. We appreciate it heaps. Hope you took something away from that session. And yeah, we look forward to seeing you guys soon and hope you have an incredible week.